Hi team, now we're going to go into a little more detail about MHC Class I. So, MHC Class I is found on all nucleated cells. So that means that any cell with a nucleus also has a Class I receptor on it. And you want to remember that these MHC Class I are on all of these different cells really to signal to the immune response or the immune system that that cell is infected. Because remember that any cell of your body can become infected, so we need a way to signal to the immune response that there is something foreign and harmful inside the cell. So here I've shown you a lung cell, which we usually draw as a square instead of a circle. Any kind of epithelial, endothelial cell is usually drawn this way. But just remember that this could also be a dendritic cell, it could be a killer T cell, it could be a B cell, it could also be a skin cell or a liver cell. Any kind of cell has the potential of being infected, and so it will have an MHC Class I on it. So MHC in general are receptors that antigen is going to be loaded into, and again, that antigen is loaded into the receptor so that it can signal to other cells that there's something going on here and we need an immune response to be activated. So in this scenario, we're going to go back to our trusty bacterium that I have down here. That's going to have a flagella on it, and that's going to be our PAMP. And remember that the antigen is just a little sequence in that entire larger structure. So over here, I've taken us through all of the different steps of what happens when a cell gets infected and how the antigen is loaded onto the MHC Class I within this cell. And there's actually more details to this process than we're going to go over in this class. So just kind of know the ones that I'm going to present here. So first we see that our bacterium is going to enter in through the cell. And I've drawn in the entering through the cell membrane as a little dashed line there. There are different ways that bacteria can enter into cells. We're not going to really get into those details, but I can point you to some literature if you want to read more. But just know that in general, a bacteria can bud in to the cell very similar to what a virus does via receptors, but it's a pretty complicated process, so we're not going to go over it. Anyway, just know that the bacterium can enter into the cell. These are called intracellular bacteria. That means that these bacteria, they have to enter into a cell in order to make more of itself. So that's what I've shown here. Step number 2, let me try to move this over a little bit more. Oh, there we go. So step number 2 is that the bacteria has entered into the cell. It's now in a little compartment, but it's going to break free of that compartment. And step number 3, or right before step number 3, let me see if I can point to it. Oops, not quite. Anyway, so you can see that once it enters into the cell, the bacterium is going to make more of itself. And I've only drawn 3 here, but remember that bacteria can replicate very quickly. So over the course of hours, you're going to have a huge number of new bacteria that can be made inside the cell. Really important to remember. So step 3, we have your own cell, any cell, so lung cell, skin cell, can actually detect if there's a pathogen inside of it. And this is also more complicated than I presented here, but we're not going to go into all of the details. But this is all part of molecular innate immunology, which we study here in the lab. We're not going to get into it. It's all sorts of cell signaling. But just know that the bacterium and other pathogens can be detected inside your cell by different molecules inside the cell. And so what happens after your own cell detects that there's a foreign and harmful pathogen inside the cell, it breaks it down, as I've shown in step number 3, and one piece of those antigens can be taken up to the surface, and loaded into our MHC Class I receptor that I've shown on the surface of the cell. So you might be thinking, oh, so our own cell can just get rid of the bacteria by itself in this process. But that's not, sorry this keeps moving so fast, that's not actually the case, because I want you to remember that this bacterium is dividing really fast, viruses too. So the process of their replication outnumbers how we detect the pathogen inside our cell and how fast we can break it down. So that's why we still need to get rid of this infected cell. The immunity that's inside your cell is not efficient enough to get rid of the entire infection for the most part. Okay, so step number 4 is that the cell loads a little piece of the pathogen into the MHC Class II. So now we're getting back to the other surface of the cell and what's happening at the top here. So now we're going to have, let's see if I can back out a little. So now we're going to have our cytotoxic lymphocyte, or our killer T cell, which is roaming around, and through all those steps that we talked about in the other videos, that killer T cell is activated, it's ready to go, it's ready to find its specific pathogen and antigen combination. So in step number 5, our specific killer T cell and its specific T cell receptor, or TCL, it finds its very specific antigen plus MHC Class I combination. So remember, that T cell receptor is very specific for our antigen. So I've labeled it antigen number 1 in this scenario. So yes, so step number 5, we have killer T cell binding to the killer T cell binding to the antigen 1 plus MHC Class I combination. And once those two things bind together, we have step number 6, which is, this is the kill signal. So the CTL is gonna get the kill signal and it's gonna initiate apoptosis in the infected cell. And we're not gonna go over how the CTL does that, just knows that that's its big job, is to initiate apoptosis in the infected cell. So that in general is all about the MHC Class I. The big picture to remember is that this is found on all nucleated cells. So lung cells, immune cells, skin cells, liver cells, everything has an MHC Class I. And this is because we want a way for our immune response to detect if cells are infected and to get rid of those cells if necessary. The other thing to remember is that our own cells, even non-immune cells, are able to detect pathogens within them and break up some of those pathogens so that it can take a little piece of the antigen and load that up onto MHC Class I. But because of the quick nature of how the bacterium and other pathogens replicate, this is not effective enough to just get rid of the infection. So that's why we still need the killer T cells to come in and get rid of that. So yeah, that's all we have for MHC Class I.